All right, shall we come back together here? All right, let's talk about the regulation of the urea cycle. So there are a few places where regulation happens. Probably the most important one is CPS1. And the first question we could ask is, what do you think would happen if you have a high ammonia concentration on that first enzyme's activity? What would you guess would happen if you have a high ammonia concentration? Yeah, it's going to activate that enzyme, right? I mean, that's going to increase the CPS1 activity. So that's just substrate driven. What effect does N-acetylglutamate have on CPS1 activity? So remember that NAG. It's essential, right? Without it, you don't have activity of CPS1. It's an allosteric activator. You need NAG to allow ATP to bind to the enzyme, CPS1. We also incidentally need magnesium, right? But we're not going to talk about the details too much. So without NAG, CPS1 can't function. So one thing to tell you is that the enzyme that makes N-acetylglutamate is also regulated by things that are related. Acetylglutamate synthetase, right? Acetylglutamate synthetase makes N-acetylglutamate. So that's kind of a little bit of an easy one. And a high arginine concentration will increase the activity of the enzyme that makes that important allosteric activator of CPS1. So we need one NAG. So allosteric means, right, you got one NAG molecule that has to bind to each CPS1 enzyme. So if we've got a lot of arginine being produced, right, if you think about that, that's the last step of the urea cycle. That's a condition under which we want that cycle to be raging, right? We want it to be going fast. We want it to be moving. So a high arginine concentration is sort of, what do they call it? It's something like, I think it's called feed-forward activation, where one of the end products is driving something that drives the first step. So
with high arginine, do you increase the synthetase one, or?
Yep, you activate that acetylglutamate synthetase, so you get more NAG molecules formed, which then is going to increase the activity of CPS1 because you've got NAG around that can allosterically activate that CPS1 enzyme. Yeah.
So the product, it makes so it's a positive feedback system?
I'm not sure exactly. Don't worry too much about, I'm not going to ask you, give me an example of that, because I don't really, I'm trying, I think it's called feed-forward activation, but it has been a while since I've thought of that term. The product of the whole cycle is stimulating something that makes the first step go faster. Yeah, I could probably confirm that would be the way you describe it. But so you get more arginine formed, you're going to activate acetylglutamate synthetase, make more NAG, which will activate CPS1. Does that make sense to everybody? Okay. Then we've got regulation at OTC, and this is simple rules, where ornithine concentration, right, and it's ornithine transcarbamylase is the enzyme. So if you've got a high concentration of ornithine, what do you think that's going to do to OTC? Substrate driven? It's going to increase its activity, right? High concentrations of ornithine are going to make OTC move. What about if you had a buildup of citrulline, which is the product of that? It's going to inhibit it, right? So that's simple. Those are simple rules, not too complicated. And then the last point, and so now we're going to build on our picture, right? We've talked about amino acid nitrogen coming in, how that gets dealt with, and urea synthesis. Now I'm going to introduce a new enzyme to you, glutaminase. We're going to talk more about glutaminase than you probably have ever wanted to talk about in your life, but it's okay, hopefully. Urea cycle enzymes and hepatic glutaminase are active at the same time, so they're coordinately regulated. When the urea cycle is cranking, so is hepatic glutaminase, and I'm going to show you why that is in a moment here. And when you got a high protein intake and or the fed state, but that contains protein, you're going to have increased activity. And the why would that be, I'll show you in just a moment, because it's an important combined effect of essentially it's dealing with amino nitrogen in a very efficient way, okay? But I'll explain more in a moment. So I guess the first thing I have here is, why do I have this? I am going to show, pardon me. I have to remember why I have this. Do I have something else? Yeah, I guess I have put this in there, I guess, to show you about arginine being the last step, in case we needed to remember that. I don't know. I'm going to leave that. Okay, let's first think about what happens if you had a defect in the urea cycle, okay, and then we'll get back to glutaminase in just a moment. So take a moment amongst yourselves, and I think we've got a few questions here, right? We've got what happens if a defect occurs in any of the urea cycle enzymes, and then what's the direct metabolic consequence of this? So take a moment and pair up and just think about this, okay? How might you answer this? Are we still talking about the questions, or are we talking about MLK Day? Are we ready to maybe come back together here? Did you nearly spill your coffee? No. Okay, good. That's good. That's good. Okay, so can we answer these questions? Whoa, what's that? Oh, wait. Yep. I'm just making sure I'm green, so I'm good. Okay, so what happens, do you think, if you get a defect in any of the urea cycle enzymes? It's possible. We have inborn errors in metabolism where various enzymes within the urea cycle or urea synthetic pathway are blocked or are not functional, right? So what do you think is going to happen? Okay, well, that would be possible, but probably something else will happen that will prevent us from having that happen. So, did anyone else what else did people talk about? Okay, what would happen if the process stops, right? So the process stops. That's often what happens because you got a cycle, right? Then you got ammonia doing what? What's going to happen to the ammonia? Build up, right? Right. Okay, so ammonia builds up and you die ultimately. It's true. Sadly, it's true. Okay, how do we know this, right? How do we know this? MNT, we haven't gotten there yet, right? We're going to talk about liver failure, right? So ammonia, I've told you, is toxic, right? What is it particularly toxic to? What in our body is probably the most sensitive tissue in the body that, if you had you think about toxic substances like, drugs and alcohol, right? Your brain, right? Your brain is very sensitive to toxic substances. So ammonia is toxic. The brain is particularly sensitive. The direct metabolic consequence of ammonia building up, or the physiologic consequence, is you've got an increased blood concentration of ammonia. We get a coma. We go into a coma, essentially. If you've got a high amount of ammonia in your bloodstream, you will go into a coma and you will die if we don't treat it. So, and we know this because when people have liver failure, we are all about managing that blood ammonia. And we're going to talk a lot, M&T gets to learn more about that. If you don't, so we have to correct it. And there are ways, we treat people that have inborn errors so that we can help manage that. Yeah?
Does it all have to do with anything like pH, drive, or whatever, being the bloodstream?
There is a pH element, but it is not only about that. Ammonia is just something we don't want much of in the blood at all, really. It's why we move it around. Remember what I told you is what we move it around as in the body? Ammonia gets moved around, as urea, right? We're gonna package it and get rid of it, but we also move it around in a non-toxic way as glutamine, right? Yeah, Dan.
It's just for curiosity, what are symptoms of ammonia buildup? So it wouldn't just be like coma, right?
You would begin by having a diminished, attention span. You would not be able to think well. You would begin to have confused thinking, get into a somnolent state. There are grades for it. We'll talk a lot about it in M&T, so I'm going to have you hold that. But it's a gradual process, right? You don't just immediately fall into a coma. It's over time. It builds up and can cause those symptoms. And we'll talk more about all of your systems get impacted, not just your brain, okay? So you get all sorts of other organ damage as well. Okay. It did it again to me. Let's see. What do I have to do again? I have to, when it's, hey, Dr. Chen. Sorry. Hey, Dr. Chen. Sorry about that. Oh, I did it. I don't know what I did. I just magically, yeah. So silly. Okay. Thank you. So the next question is how does amino acid nitrogen feed into the cycle? Okay, I've given you the first few bits of information around that, right? Got transamination reactions and getting amino nitrogen into glutamate, but we're going to, up here, no, I'm not up there. Gee, why do I have that? Oh, over here. Sorry, my animations are a little bit. So let's start over here. We talked about aspartate, right? And this is part of the cycle. But we have quite a lot of alanine coming in from the intestine, actually. And we're going to talk next about the intestine because we're still in the fed state, right? We're thinking about the fed state. So alanine comes in from the intestine through the portal vein from glutamine metabolism. And glutamine metabolism is very important to the small intestine, just very important. So that's one way that we can easily get other amino nitrogen coming into the cycle is through ALT and then into the AST reaction. Incidentally, we can have other amino nitrogen feeding in through other transamination reactions as well, okay? So that's how we get some of the amino nitrogen in. And now we're over here, okay? Up here, we've already talked about how we have transamination reactions feeding into glutamate dehydrogenase releasing ammonia. We also have glutamine that is available, and we're going to talk about there are different sets of cells that are in the liver potentially contributing. So we're moving a little, we're going to be broadening the scope a little bit so that we're going to talk about different cells in the liver, okay, beyond the periportal hepatocytes in a moment here, and I'll show you that in a diagram. So there are lots of ways that amino nitrogen is feeding into this urea synthetic pathway. So this is what we're going to call the intercellular glutamine cycle in the liver. And, we had talked mostly about the periportal hepatocytes, right? Coming in from the portal circulation from the intestine, where we've got this amino nitrogen feeding into glutamate, getting ammonia into the urea cycle. Well, incidentally, in the blood, coming from the blood, we can get glutamine coming in, okay? Also, glutamine, is also coming in from the intestine. Whatever the intestine doesn't degrade, it doesn't take all of the glutamine, right? So we have glutaminase enzyme that is basically breaking it down to glutamate and ammonia, and then the glutamate undergoes the dehydrogenase second step reaction, which I didn't show the GDH, I don't think, here. And then further down the pike, I guess you'll say, and it's separated in distance, not shown here obviously very well, is around a different zone in the liver, is near the central vein, okay? And it's sort of where we're leaving. Circulation starts, comes in from the intestine in the portal vein, and then it leaves via the central vein. So paravenous hepatocytes further down the line are around the central vein as we're leaving the liver, right? So essentially, what's happening in these paravenous hepatocytes is that any ammonia that is coming from the bloodstream, of course all the blood is coming through circulation, right? And there can be some ammonia potentially that might escape, getting incorporated into the urea synthetic pathway. That ammonia is going to be taken up by these paravenous hepatocytes and acted on by this enzyme glutamine synthetase, okay, where you're essentially making glutamine out of it. So remember I told you ammonia traveling around tissue to tissue, it's toxic, right? So we make glutamine. A lot of tissues can make glutamine, have the enzyme glutamine synthetase, so they can synthesize glutamine. And this is the liver's way of having a backup plan, really. It's a backup system for dealing with or detoxifying ammonia. So it's an important thing that, lots of it, a lot of that glutamine and amino nitrogen is going to be dealt with very efficiently, especially in the fed state by the urea cycle. But any ammonia that happens to escape is going to be detoxified by being made into glutamine right before that blood leaves the liver, because it goes through those paravenous hepatocytes. So, liver has two ways of dealing with amino nitrogen, right? One, it synthesizes urea. The other is that it can synthesize glutamine. It's a different set of cells that does that. And I guess that's all I'll say about that. Is there a question? And notice this, right? Magnesium is there because it cleaves one ATP. It takes an ATP to make glutamine from ammonia and glutamate. You make glutamine, and that glutamine then can go out to be taken up by other tissues, including the intestine and whoever else wants it. Any questions at this moment? I think I have a summary here. So glutaminase action is happening, as we said, in the periportal hepatocytes. And that's important because it's feeding that ammonia directly into the urea cycle, right? And then in the paravenous hepatocytes, we are synthesizing glutamine from any ammonia that escapes getting into the urea cycle. And under conditions where the urea cycle is not very active, that's what we do with it, right? Because it's not always active, right? In the fasted state, for example, urea cycle is not going to be going very well, right? So any amino acid nitrogen or ammonia that's coming in, it's important that we can detoxify it under those conditions. So in those conditions, it's the paravenous hepatocytes that take it up and use it very, very well. And in fact, this enzyme, glutamine synthetase, has a very high affinity for ammonia, right? High affinity means low Km. I don't know how your brain works, but anyway, I have to think about that. So high affinity means, it's not a large capacity, but any ammonia that is even in the vicinity of a glutamine synthetase enzyme, it's going to suck it up and make it into glutamine, right? So that's important. It's a great detoxifying mechanism. So, how would you answer this? How does the location of these two enzymes, the fact is, glutaminase is in the periportal hepatocytes, glutamine synthetase is in the paravenous hepatocytes, and they are separated. We'll just say they're, miles apart. Not miles, but they're far apart. So how would you answer that question? I'm going to take a moment and talk to your neighbor. Articulate it to your neighbor, right? Take 30 seconds, please. We ready? Was that enough time? So who came up with something? What would you say? Why would that be a good thing? And you know, there are lots of examples of this. Dylan?
Because of the affinity for ammonia, so if they were in the same location, that the affinity would be, it wouldn't take as much for the glutamine to be created, and your recycle would happen as readily.
Okay. You're along those lines. You're thinking right. What's kind of what would happen if they were in the, let's just think about that, right? If they're in the same place, what would you be doing? What would the cell be doing? You got both things going on at the same time. What are you doing? Yeah, and you're competing, right? Well, what are you? You're not really. You would. Someone tell me. You know what does? Yeah, and Dylan, you just said it a little bit. What would you add to that?
Glutamate is breaking down glutamine, and then you're actually making glutamine.
Yeah.
So you'd be kind of like competing, you'd just be kind of self-competing.
Yep. And what does it take to make glutamine? ATP, energy, right? So does the body like to conserve energy? Yes, right? I mean, because you know we're not an endless supply. We don't have an endless supply of food and all those things, right? So we have evolved, right, to not waste energy if we can avoid it. Hence the couch potato. No, I'm just kidding. So if you had it in the same thing, it's that the potentially futile cycle. Maybe that was the thing that threw us off there because it's sort of leading. That's what you would have, right? You would just be using energy to make glutamine, then you'd be breaking it down. Then you'd be using energy, you know, so you'd just be burning a lot of energy for no purpose, right? And that's what a potentially, a futile cycle is. And we have examples of that throughout biochemistry, right, or metabolism. Okay. So it's useful that they're in separate cells, and this is just showing you the basic unit of the liver is highly, highly organized. All of these are hepatocytes here. We've got branches of the various vessels, portal vein, central vein, right? And here's a you know, fairly obviously not drawn to scale, but you can kind of see that portal hepatocyte around the portal vein, and then ultimately going through lots of this, you know, highly architectured, structured sequence and circulation. You end up leaving the liver at the central vein, and thus, you know, any ammonia that doesn't get captured and made into urea here is going to be made into glutamine before it leaves the liver. So that's the intercellular glutamine cycle. And I've got some slides that I'm going to just skip through because I've just written them out again. And I don't think there are any new concepts on these. It's just to help you see them separately, what they look like, the structures. And just remembering that glutamine, that's what it looks like, and it's got glutamine in the name of it, so you can keep it straight, right? I don't want any of you to get those mixed up. Glutamate is the thing that's involved in all those transamination reactions, and glutamine is the one that's carrying amino nitrogen around between tissues. And then we had talked about glutamine synthetase having a high affinity, low Km for ammonia. And that's an important thing. If you want to write it here as well, that's probably a useful thing. Okay, lastly, I think this is somewhat lastly for the liver, is what happens to the carbon skeleton, right? So we've just talked about what happens to the nitrogen. And the fate of an amino acid now is going to vary depending on the physiologic state, right? Whether you're in the fed or the fasted state. And this diagram is a little busy, and it's already, I'm going to try to walk us through it in somewhat of a methodical way. So staying in the fed state for a moment, you can ask first, is carbohydrate energy intake adequate? Thinking about protein sparing, right? Carb and fat. Actually, I could have put that in there as well, right? So, and particularly carb, because what does carbohydrate trigger in terms of hormonal milieu, right? You take in a carbohydrate, what do you get? Insulin, right? Yeah, that's why I put carbohydrate there. So if the answer to that is yes, right, we got insulin is high, glucagon's low. If amino acids, are they in excessive needs for protein synthesis? Okay, so they're not, if they're not, well, okay, we're going to take whatever we need to and we're going to make proteins. The liver has a certain need for its own protein synthesis, and it will allow some of them to go on to other tissues or whatever. But we'll take care of the liver's needs because it's got first pass at everything. Yes, if the answer is yes, that we've taken care of all the needs for protein synthesis in the liver, then we're going to catabolize amino acids. We're going to feed the amino nitrogen into urea, right? So in the fed state, we're really, predominant form of nitrogen excretion in the fed state is as urea, right? And we're going to talk about differences in the fasted state in a moment. The alpha keto acid now, the carbon skeleton, could be used for energy. Mostly it's probably going to be used for fatty acid synthesis. Some of it does get through to be used to make glucose, but not, and it's not a direct, not a predominance. It's as glycogen via gluconeogenesis. Sorry, I misspoke there. It's glycogen from the carbon skeleton, but a lot of it's going to be stored as fat in excess or catabolized completely for energy as needed. Over here in the fasted state, you got the hormonal milieu is the opposite of the fed state, right? And under these conditions, I've tried to make this arrow, this is intentionally smaller, so just, I'll point that out so that you can appreciate that under these conditions, we're going to be excreting less as urea. We're actually, I'm going to talk about this, but we'll be excreting more at the kidney as ammonia. Okay, I'm going to hold that thought. We're going to get back to that. We're going to talk about fasted state and what's happening at the kidneys in a moment. But we're going to be making glutamine at the liver. The liver will be making more glutamine proportionally under these conditions. Why? Because the urea cycle is not really moving, right? It's not moving, so that ammonia gets taken up by the glutamine synthetase enzyme in the paravenous hepatocytes. We make glutamine to ship out to other tissues. And especially the intestine really appreciates that glutamine coming from the liver, right? Because the intestine relies on glutamine for energy. So when you're in a fast, that's a really important source of energy for your intestine, okay, is the liver synthesized glutamine. Incidentally, this is also happening if you're going to be using amino acid carbon in this pathway when you don't have adequate carbohydrates. So if you're on a really low carb diet, right, 20 grams a day or something like that, like the Atkins has you do, you're going to be using alpha keto acids for making glucose, big important thing that we do with that, to feed the brain and other obligate glucose users. And we'll make ketones out of it as well. And the amino acids in this case, where are they coming from mostly? In the fasted state, where are we getting amino acids from? You guys know this. Say it. Where? Where is it coming from? Sorry? Say it louder. Catabolism of what, predominantly? Proteins coming from mostly what tissue, would you say? Muscle, right? Muscle is our best reservoir. Thank you, Sam. Right? So in this case, it's coming in through the diet. In this case, it's coming mostly from skeletal muscle. So we'll talk more about that. What else do I want to say? Let's just look at that here. I guess that's it. So I'm just reminding you here, I guess I've written it here now, leucine is uniquely ketogenic. Again, how would you explain that? Take 30 seconds, say it to your neighbor, articulate that. Explain that to each other. Why is it that leucine is uniquely ketogenic? What does that mean to you? Take 30 seconds. Is anyone ready to articulate it? Can I get somebody to help us out here? So how would you say that? What is it? How do you answer that question? Why is leucine uniquely ketogenic? It doesn't have to be perfect. I'm not the most articulate, especially somehow this morning. Okay, so Laura is helping us out here. It yields just 2 carbon intermediates, right? But a little more on that, right? Why is it that it's, so when we say uniquely ketogenic, producing 2 carbon units, what else? What's the other part of the explanation there? What is that uniquely ketogenic? Why is it not glucogenic? Because when we're saying it's uniquely ketogenic, we are saying it's not glucogenic, right? Which means what? Help me out. Yeah, Lexi. Can't be used to make glucose. It can be used to make fat. All amino acid carbon can be used to make fat, right? But because it yields just 2 carbon units, right, it's not anaplerotic. It does not contribute carbon to the TCA cycle, right? And here we are over here, and we have to, if we can't contribute carbon to this cycle, then we can't keep gluconeogenesis going, right? We have to have carbon coming in. So uniquely ketogenic means it can't do that. Okay, last slide here. We had talked about in the fed state, 60% of the amino acids coming into the liver get catabolized, right? So still talking about the fed state here, what happens to those amino acids, which ones are being released unscathed? And I told you at the beginning, and leucine is one of them. Branch chain amino acids, right, are the ones predominantly that are being released in bulk. They don't undergo catabolism at the liver as the first pass. They get allowed to pass through. 70% or so of the ones leaving the liver are branch chain amino acids, so it's a vast majority. And then I've got some other, we're going to use 20% for protein synthesis. We're going to talk more about the branch chains in just a second. Which amino acid is highly conserved, meaning it doesn't go through transamination as the first, second, third, fourth, or fifth step. I mean, it's way down the line. Do you remember? Lysine, it was. Lysine. And again, doesn't, so it stays around. And I'll just give you this, but we're going to talk about it more when we get to the muscle, okay? But branch chain amino acids, as they leave the liver, they are going to be taken up by the skeletal muscle in large part. And part of the reason for this is that the enzyme that acts on branch chain amino acids, it is a transaminase, but it is specific for branch chain amino acids, and it has really low activity in the liver. So that allows for that to happen. Okay? So, these little details, I'm hoping, will help you put it together of, why it is. It makes sense. Biochemistry is fun in that way, that it makes sense. And I know I'm a geek for admitting that I like, the urea cycle and transamination and all that. But Hopefully that makes sense to you, right? So if the enzyme that acts on branched chain amino acids, the transaminase, is not active at the liver, that's how it allows it to pass through those branched chain amino acids. So after a meal, most other amino acids remain relatively constant in the blood, actually, because the liver is pretty good at taking them out. Branched chain amino acid concentration goes up a lot after a meal. So in the blood, you'll see branched chain amino acid concentrations going up. Okay, any questions about what's happening at the liver? And what time do we stop again? 35, okay. No questions? I've stunned you into silence, hopefully not. All right, shall we talk about the intestine? Oh my, Bronwyn again at work. Boy, and that's slow. Okay, I should specify that we are talking about the small intestine, okay? The intestine where all of the fun happens in terms of digestion and absorption. And all right, so we're going to talk about a few things at the intestine. A lot about glutamine and the enzymes that are involved in glutamine metabolism, and then more about glutamine. Then we're going to talk about how this interesting inter-organ cooperativity happens to help us facilitate whole body protein synthesis. Okay, so we'll hold that thought and get back to it in just a moment. So let's start with our little very basic diagram here. So we're going to stay talking about fed state right after a meal. Most but not all amino acids leave the intestine. So we had talked about how so many will go, through the portal vein to the liver, and we've just talked about what happens there. But let's talk about what happens to the amino acids that the intestine preferentially uses, because the intestine needs particularly glutamine. So first question is, why does the intestine need to retain some of the absorbed amino acids, right? And the answer to that is that it has its own maintenance needs, right? So lots of structural maintenance and apoproteins, the one that is needed for chylomicrons. Do you remember to make chylomicrons, which apoprotein we need? Right, what is it, ApoB48? Is that what it's called? Yeah, ApoB48. I had to remind myself of that too. Digestive enzymes, gut hormones, etc., right? So those are important. The other thing that it retains some, and again preferentially, particularly glutamine, is energy needs. So the intestine has a fairly high energy requirement and really needs glutamine for energy. So primary energy source, much of it will come in through the diet. Proteins tend to be rich in glutamine, most proteins. And it also comes in from the blood. And if you think about that, we've talked about glutamine being the very important carrier of amino nitrogen between tissues. Well, it happens to be the most abundant amino acid in blood, actually, for that reason, right? Because all the different amino acid pools that are undergoing metabolism in the various tissues, oftentimes we're producing glutamine to get that amino nitrogen out. Blood glutamine is going to come from muscle in large part. Skeletal muscle is huge, and we're going to talk about how branched chain amino acid metabolism produces glutamine. So the muscle is a major supplier of glutamine to the intestine. Also the lungs, and then the liver, and which cells are making glutamine at the liver, which type of liver cells. Perivenous hepatocytes, right? Again, because they're the ones dealing with that ammonia. So let me just see how I want to tell you this. So in terms of in the fed state, we'll just say, only about 20% of the intestine's energy comes from glucose. So you've got 40% coming in from the blood, 40% coming in from diet, and glutamine is primary of that. The intestine uses asparagine too. That's the cousin of glutamine, right? But for our purposes, you can just kind of think of it as glutamine, right? Intestine using it for energy. The impact that this has is a trophic effect on the GI mucosa. So just being able to provide that energy that the intestine needs to have the rapid turnover that the intestinal cells do, that supply of glutamine is very important to the intestine so that it has, we can proliferate the cells. And then I want you to think about what enzymes does the intestine need to metabolize glutamine, and I'm going to show you, okay? So you can answer that question in just a moment here. All right, so let me think about how I want to orient you to my somewhat busy, colorful slides, but you know, that has a rhyme. I do have a little bit of rhyme to my reason, or reason to my rhyme, or whatever. So the purple circles, wherever you see that, that means compounds that are produced from glutamine metabolism. If you see a sunshine, that means that's going out from the intestine. Those are export products, okay? So glutamine, of course, you could have guessed this, right? I mean, I could have left this blank and you could have filled it in. That glutamine, the first thing that the intestine is going to do with that glutamine, it's going to catabolize it using glutaminase to yield glutamate, right? And then we've got ammonia, and remember, that ammonia is going through the portal vein to the liver, and what happens to it at the liver if you're getting ammonia coming in? Portal vein, periportal hepatocytes in the fed state. What are we going to do with that ammonia? Urea, right? Going to make urea under those conditions. All right? If it were the fasted state, we'd probably be making it into glutamine at the liver, actually, but we're talking in the fed state. What else is happening? Okay, so we just had a question. I've forgotten. Katarina, you were asking this, right? Where do we get ornithine from? Well, we can get it in foods too, okay? And there are other ways of doing it, but we can make it. Ornithine we make from glutamate, actually. And now I've got to just remind myself so I don't misspeak here. Yeah, so we can use glutamate. What these little dotted lines are is what we use glutamate for, and these are just a few of the things. Glutamate is a highly useful non-essential amino acid because we can make another non-essential from it. We can make proline, right? And we can make this glutathione is a peptide. It's a tripeptide that is very important for antioxidant activity in the cell. Glutathione peroxidase, you ever heard of that? Breaks down peroxides, which are really dangerous to cells, actually breaks them up. It's what we use, our immune cells release peroxide, right? Hydrogen, we've all had a cut, right? And you put hydrogen peroxide on it, and you know what it happens is all bubbles and all that, right? Our immune cells, especially neutrophils, some of our first-line defense mechanisms, release hydrogen peroxide, right? Which is in the face of a bacteria, bacterium will kill the bacteria, bacterium. Then we need a way to deal with that, right? So glutathione peroxidase breaks down the peroxide, the hydrogen peroxide, to kind of disarm it, right? It's an important thing to have. So glutamate, you can see how important it is. The other thing is that it can be used to make ornithine. Ornithine then can be used to make citrulline. Now, actually I'm going to hold that thought because I think that's a useful breakout question to ask you. So we'll leave that over here for a moment. And citrulline goes out. That's an important thing, and there are some enzymes that are involved there. The other thing that happens is glutamate will undergo transamination with alanine transaminase to make alanine. And so alanine is both produced and released. Alanine gets taken up by the liver. All right, so the major export products of glutamine metabolism are these three: ammonia, alanine, and citrulline. Now, take a moment between yourselves, and I want you to look back, if you have to, at your slides, and I want you to talk to each other and you tell me which enzymes are needed here in the intestine to convert ornithine to citrulline. And we have 1 minute, don't we? 1 minute. Can you take 1 minute and just see if you can think about this? And maybe beginning of next time we'll actually discuss it. How about that? Did I forget to turn that off? Turn that on?
no.
Oh, I think I did. I may have cut the recording short.